my great pleasure to introduce our first speaker, Dr. Jeffrey Cummings. There are many ways that I could introduce Dr. Cummings. He is my mentor for my years at UCLA. He's now the founding director of the Cleveland Clinic Lou Ruvo Center for Brain Health. He's the Camille and Larry Ruvo Chair for Brain Health. He's also a professor of medicine at Case Western University and adjunct professor of medicine in psychology and neurology at University of Nevada, Las Vegas. This year, he was awarded the Bent Wingblad Lifetime Achievement Award at the Alzheimer's Association International Conference. A few years ago, he was deemed by the Journal of Alzheimer's Disease as the 6th most prolific Alzheimer's researcher in the world. And recently, he has devoted much of his energy to leading national and international clinical trials of extremely promising therapies for Alzheimer's disease. I've invited him to set the stage for our entire day of discussion of clinical trials by telling us how we got here in the state of Alzheimer's disease clinical trials. So please welcome Dr. Jeffrey Cummings.
Thanks, Josh, and good morning, everyone. Great to be here. UCI is one of my very favorite places, and I'm delighted to be here. Frank is one of the world leaders in Alzheimer's disease, whose work I have always admired. So it's a pleasure to be here and to be part of this program. And the rest of the speakers today are terrific. I'm so looking forward to learning from them and getting the update on what their work has brought to them. So I was really happy when Josh invited me to this. Josh is one of my greatest accomplishments in the Alzheimer's disease field, so I take special pride in that. And I have been deeply involved in clinical trials and recently thinking about, well, how do we learn more from them? And so when Josh said, well, why don't you review the history of clinical trials and put the lessons from them in that perspective. So that's what I've tried to do, and I'm looking forward to discussing this with you also. Here are my disclosures. I work with a lot of companies. If you do clinical trials, you wind up working with a lot of companies. So we're trying to find the best possible therapies, and these are the companies that I worked with to try to do that. So I want to start with the idea that this is an idiosyncratic history. This is my history written for you. And of course, history is always written in the present about the past. It's not like we just review the history, right? We create the history. And so I'm going to create the history as I see it for the most important and informative agents. This is not a comprehensive history of every drug which has been tested for Alzheimer's disease. This is a history of the agents that I think we learned most from. So I admit to the idiosyncrasies, and if you want to challenge them in the question and answer period, I'd be delighted with that. And if I didn't get your favorite agent into this history, why, speak up for it and champion it in the discussion period. I'll start with the cholinesterase inhibitors and memantine. I will also talk about monoclonal antibodies. This is an area where we are making, I think, a lot of progress. Small molecules means pills. So I'll talk about the pills that have been used and where we are with pill development. I'll talk about the progressive refinement of trials. You'll see the kind of trial we do now is quite different from the kind of trial that we did in the past. And try to derive at the end some lessons. What have we learned from this? And I couldn't echo Josh's comments more about the importance of trials. This is one of the few places where the only kind of data that the FDA will accept for approval of a drug is from a clinical trial. And that means no trials, no drugs. So if we don't do it now, we will never have the drugs that we need. So this is a unique circumstance in the development of science where everything funnels down to one thing, doing and winning a clinical trial. And we feel a great responsibility to all of you who participate in clinical trials. It's our ethical and moral obligation to make sure that we learn from every trial, that we take as much away from that trial as we can. That patient, that caregiver, have put that patient's life at risk because by definition, a clinical trial is done because you do not understand that drug. So you do not understand its efficacy and you do not understand its safety. We do everything we can to ensure that patient's safety. Nevertheless, we understand there's risk and we honor the risk that our patients put into those clinical trials. That's why lessons are so important. Well, here are my thoughts. I'm going to start off with a drug called tacrine in 1993. And I'm going to tell you that this is the breakthrough agent. So it started off with a kind of odd history. The New England Journal, one of the most prestigious journals in the medical profession, published this article, Oral Tetrahydroaminoacridine, Long-Term Treatment of Senile Dementia. How long has it been since you've seen the term senile dementia? We would never use that now, right? Alzheimer's type. So this was 86. Marked improvement was reported by Bill Summers. But the instruments that he used were unknown. The trial methods were odd. When challenged by the FDA, the records were lost. And so the trial was in substantial jeopardy. But there was just enough there, and there was just enough frustration in the Alzheimer community because there was no therapy, that the advocacy groups, the Alzheimer's Association, many others said, we want to consider this more seriously. And it's a demonstration of the power of advocacy because this drug, really the trial was so questionable that if it hadn't been for advocacy, it would not have been taken forward. But it was taken forward and it was shown to be an efficacious agent. Here were two trials, both published in 1992, a double-blind, placebo-controlled, multi-center study of tacrine for Alzheimer's disease led by Ken Davis. And Mary Sano is here today. She's going to give one of our lectures. I don't see Mary right now, but there she is right here. So Mary works with Ken now and has worked with him for a long period of time. The other one, published in JAMA in 1992, a controlled trial of tacrine in Alzheimer's disease led by Marty Farlow. Marty's still very active in this field, and I'm in touch with him frequently, including this week. So these were two formative papers that said, yes, tacrine actually does improve cognition in Alzheimer's disease. And it defined many of the assessments that we still use today. That's one of the reasons I think it's a breakthrough agent and why I consider it so formative to the field. So the cognitive outcome was something called the Alzheimer's Disease Assessment Cognitive Subscale, the ADAS-COG. And you're going to see that the latest trials that I present today are using the ADAS-COG as an outcome. So from first to last, we are using the ADAS-COG. The global outcome was the Clinical Global Impression of Change, the CGIC. We still use a version of this. The behavioral outcome was called the ADAS non-COG. Now, almost no one has heard about the ADAS non-COG, and for very good reasons. Because it added everything that is not cognition into one scale. So it had depression and it had tremor. Well, what kind of scale has depression and tremor in the same scale? Very difficult to do. It did open the opportunity for other behavioral instruments to be developed, and I helped with that. Study entry was done by the Mini Mental Status Examination. How many of you heard about the Mini Mental Status Examination? Yeah, everybody? Yeah. So we're still using it, invented in 1975, used in this study as 10 to 26, still a common range for mild to moderate Alzheimer's disease. So you see the principles that we're still using today embedded in these tacrine trials. So it was approved by the FDA in 1993. The head of the neurology division at the time was Paul Lieber. And Paul wrote in 1990, and I've shown you these. I thought it was kind of cool. I went back and pulled them out. You can see they were done on a typewriter, right? 1990, it's done on a typewriter. So Paul wrote this, what were called the draft guidelines, and amazingly, they were actually never themselves updated. There have been many guidelines since, but this original definitional set was never redone, and they're brilliant. Paul was a great and deep thinker, not a person who lacked self-confidence and was able to present his ideas strongly. And he divided them into symptomatic and definitive treatments. Now we would say symptomatic and disease-modifying. He said trials must have dual outcomes. The central problem of Alzheimer's disease, which is memory, so they must have a cognitive measure, and they must have a measure that implies clinical meaningfulness. That is, either they improve on function or they improve on the global scale. Because if you can just move a little memory scale, a little tiny bit, and you don't actually impact the patient's lives, that's probably not a very important drug. So Paul said, you've got to do both. You've got to move cognition, and you've got to move something about the patient's life to make it meaningful. And we still live with these principles, although as we work with earlier and earlier patients, or even people at risk for the disease, of course, we've had to modify these. And there's a long discussion in this paper about placebo comparisons and the importance of internal controls versus historical controls. So internal controls is a placebo group. We believe very strongly that that is the way to get valid data. And again, we understand the challenges that raises. So this was fantastic because it said in 1993 with the FDA, Alzheimer's disease is treatable. And that changed the world. It meant that for the first time, a doctor could sit with a patient and say, I have something to offer you. And until then, all you could say was, if you get depressed, I have an antidepressant, and if you have a behavioral problem, I have an antipsychotic. That was all you had. So all of a sudden, you had something to try, and that changed the kind of conversation, and it changed the history. We're waiting for that next step now for even more efficacious agents. But this was a pivotal moment in the history of Alzheimer's disease. Now, it was a terrible drug. It produced liver toxicity in 50% of the people who took it. I had a nurse whose only job was to follow the liver functions of my patients. Her only job, because we had to be on top of them so regularly. You had to take the drug 4 times a day. So in reality, if you could remember that, you didn't even need the drug. So these were challenges which were very difficult. And they led then to the next step, which was the approval of the 3 drugs, the 3 types of cholinesterase inhibitors that we have today, which were donepezil in '96, rivastigmine in 2000, and galantamine in 2004. So these are the drugs that are still in our repertoire now. The donepezil story started off at 5 and 10 milligrams, approved in 1996. And then people said, well, couldn't we give more? And turns out we could. If you push to a higher dose, you get better efficacy and you get more side effects. So it's a very tricky dose to use, but we do sometimes push to the 23 milligram dose approved in 2010. Rivastigmine, or Exelon, started out as an oral form, approved in 2000. And then the patch, which turned out to be a great way of reducing side effects, was approved in 2007. So that has been a very important advance in terms of side effect management and was a great lesson. And then the high-dose patch was approved in 2012, again saying, well, if we're getting this much efficacy, can we crank up the dose? Can we get better efficacy with higher dose? And we can, again, at the expense of higher side effects. Galantamine had its own interesting history. It came out as Remenil in 2004. Turns out doctors confused that name with a cardiac medication that was similar, and so the FDA made them change the name to Razidine in 2005. So this is just a little kind of nuance in the history about how drugs can change names even after approval if there's some sort of marketplace mischief that they get involved with. So donepezil and rivastigmine are approved for mild, moderate, and severe Alzheimer's disease, and galantamine for mild and moderate Alzheimer's disease. And these are still the drugs that we're using today. They are useful, and they're certainly better than a placebo group in almost every study, but they do not have the ability to modify the disease, and their effects are modest in magnitude. So we use these drugs. We use them in essentially all of our patients, but they're not the drugs that we want for our patients. So there were various casualties and catastrophes along the way of developing these successful drugs, cholinesterase inhibitors. For example, there were other cholinesterase inhibitors that were treated. Visostigmine had a very short half-life. I did a study with this. I had to get caregivers to give it to the patients every 2 hours. That's not a feasible solution. Metriphenate was a very promising cholinesterase inhibitor, and then a few patients developed respiratory paralysis on it. So in the middle of a trial, we took everybody off of metriphenate. It had to, and that was the right thing to do. Well, maybe if you don't inhibit the enzyme, maybe you can just directly stimulate the receptor. So muscarinic agonists were tried, and they produced too much syncope, so patients fainted when they were given xanomeline. There's a whole other part of the cholinergic system that has to do with nicotinic agents, and so these have been studied in many trials, but the outcomes have been inconsistent. So I hear today, there's been all these amyloid failures, and therefore we shouldn't be working on the amyloid hypothesis. Well, here's a lesson. Look at the cholinergic system. Look at all of the failed drugs in the cholinergic system, and then see that we had successes when we got it right. So one lesson is not every drug is going to work against a target. There's all sorts of nuances that evolve when you're working with these drugs. And so don't give up. If you've got a good idea, the science is right, keep in there with the drugs until you make them work. So those were the cholinesterase inhibitors, 1993 to 2004. And then Memantine, the other drug that we currently use a lot, it was approved in 2003. Now, I want to say something that I think is important. Memantine was the last drug that was approved for Alzheimer's disease. 2003, since we have had a new drug approved for Alzheimer's disease. It's time, right? It's time for a new drug for Alzheimer's disease. So Memantine is a different chemical class, an N-methyl D-aspartate inhibitor. It's given either as monotherapy or as add-on therapy to the cholinesterase inhibitors. It's approved for moderate to severe Alzheimer's disease, not mild to moderate, so it's a little further into the illness. And it's essentially always used in conjunction with cholinesterase inhibitors, under rare circumstances as monotherapy, but usually as add-on. And this then established a new lesson, which is that new drugs would be given on top of the standard of care. So we want to give our patients everything that's approved and then add the new drug or placebo on top of the standard of care. So the trial never compromises the best care that we can give to that patient. And then the new drug is randomized as drug or placebo on top of that existing standard of care. And that lesson emerged from our work with Memantine. So now let's switch from the cognitive enhancing agents to disease modification. That's about 75% of the drugs in the current pipeline are disease modifying agents. That is, we're not trying to just improve the symptoms, we are trying to slow the disease. And we believe that ultimately that is a better outcome, though they will be used in conjunction with drugs that can improve the symptoms. So the first drug of great magnitude was called AN 1792. And it came out of the work of the late Dale Schenk, and many of us here knew Dale and liked Dale and are sorry that he was lost to cancer recently. So here was a vaccine. And this is such a powerful idea, right? When you're a child, you get your measles vaccine and your diphtheria vaccine, and you don't worry about it for the rest of your life. Wouldn't it be great if you could get your Alzheimer vaccine and never worry about it for the rest of your life? That's ultimately what we want. That's where we're going. We're a long ways from it. It was based on an animal model of Alzheimer's disease, and a clinical trial was done, published in 2005. So this was the first major disease-modifying trial. It was the first immunotherapy trial. I'm going to show you some others. Antibodies were generated, so when they got the shot, they did generate the body's response to the amyloid protein that we think is key to Alzheimer's disease. The amyloid plaques were reduced. So this was a winning agent. It reduced those plaques. How did we know? At autopsy. Well, that suggests that the drug was not completely successful, right? If you're getting the answer at autopsy. So it was also the first time where we saw that we could remove plaques and not stop the disease. So that's been an important lesson as well, and I'll try to explain that conundrum as we go along. Unfortunately, 6% of patients in this trial developed encephalitis, and so the trial was stopped like that. As soon as we know that there is a major patient safety issue, we stop the trial. That's how we keep our patients safe. And there were no consistent clinical benefits, as I mentioned. But it was a start, and it was an important start, and it showed that we could move amyloid around. So the next trial then was tacrine, and this is a drug that taught us a lot. So this is the R enantiomer, that is one form of ibuprofen. So this is Advil, and it was known from epidemiologic data that people who take more Advil are less likely to get Alzheimer's disease. So they thought, well, let's make this a drug. It was known to have an effect on an essential enzyme, gamma secretase. So there are two enzymes that produce the amyloid protein in the brain. One is gamma secretase, one is beta secretase. We're going to talk about both of those. And so the R enantiomer, which appeared to be the effective enantiomer, was pulled out of the chemical, put into a clinical trial. And you see here now the kind of graph that we see again and again. No drug placebo difference. But why was that? Well, let's look at the lessons. Target engagement, that is the fact that was it actually inhibiting gamma secretase, never shown in humans. When they went back to look at the data, actually it had very poor penetration into the brain. You have to get the drug into the brain in order to make it work. And this has become more obvious to us recently. There was no dose response that was effectively explored. And the phase 3 trial was based on a negative phase 2 trial and a subgroup analysis. So they said, well, looks like to us that mild patients with high serum levels did the best, and so we'll do a trial of mild patients and give them higher doses. Those are logical things, but when you base phase 3 on a phase 2 subgroup, you usually cannot reproduce the results of those subgroups because the trial is not structured in that way. So all these became lessons. Let's show target engagement, let's show brain penetration, let's know the dose before we take it into phase 3, and let's not base phase 3 on a failed phase 2 trial. Those were all important lessons. We're having a little trouble learning them, but they are lessons that emerge from our experience with tacrine. So the next drug then that I want to call attention to is semaglutide. And this is the 2013 trial, a very important trial. Because the high lines, the higher you are on this graph, that means you're getting worse. And the lower you are on this graph, those patients are doing better. So you see that the low line, the patients doing best, is the placebo group. And the high lines, the patients getting worse, are the semaglutide 2 doses. This was the first trial in which we clearly, systematically made patients worse. And I think this is something very important. We've got to learn from this. We cannot have trials that make patients worse. And it was based on great biology. We knew that this enzyme would produce the amyloid protein. And yet, when we actually stopped effectively the enzyme, we made patients worse. So that tells us how much we have to learn about the biology. So this was a gamma secretase inhibitor put into mild, moderate Alzheimer's disease. Now we say Alzheimer's disease, but of course there was no biological confirmation. It was just a clinical diagnosis. We now know that we're frequently wrong, too frequently wrong. We worsened cognition, we worsened function, and we produced skin cancer in the treatment group. So these were powerful, powerful drugs. We learned a lot from them. We also learned that we need to understand this biology extremely well. Because we're putting our patients at risk, even if we think the biology is really great behind these drugs. Now, I just wanted to call attention to some of Josh's work here. Why not, since we're at UCI? So one of the things that we've become very interested in is the effects of using these drugs around the world. Because you often have to have 1,000 patients in a trial now in order to show whether the drug is working or not. Well, there are not enough centers in the United States like UCI to produce enough patients in a short enough period of time that we can do the trials exclusively in the United States. So we have trial sites all around the world when we are working on these drugs. So here I'm showing you whether patients stayed in the trial over the course of the 18-month exposure to the drug. And what you can see is, at the top line, those patients stayed in the trial. Japanese patients do what they're told, all right? They stay in the trial. The Eastern Europeans, you can't depend on them, I'm sorry to say. You see, they come right out of the trial. This is important, right? Because we've got to know how to test these drugs worldwide. So this is an important lesson. So the next drug then was bapineuzumab, 2014. This is another immunotherapy, but it's the first time we've turned away from vaccine. Remember that experience with 1792. We're not going to do that again, but maybe we can just take the antibody, make it outside of the patient, and then give it to the patient intravenously. That would be the approach. So that's how the monoclonals are developed, is to direct immunotherapy with antibodies that are raised externally. So a phase 2 showed no effect on the 8S-COG. Here's the 8S-COG, right? Here's our beautiful instrument. Post-hoc analysis. What did we learn about post-hoc analysis? It's almost never reproducible. Suggested benefit in E4 carriers. So a phase 3 trial was done and showed no benefit in either carriers or non-carriers. So you see that we're struggling a little bit to learn this lesson, right? We've done it again. These were mild to moderate Alzheimer patients, again, not biologically confirmed. At that point, we had no easy way of biological confirmation. So here's on the left is the phase 2 trial showing no drug placebo difference, but when it was looked at post hoc, according to carriers and non-carriers, it suggested that the non-carriers did better. But you see then on the right, these graphs that I'm now teaching you are so common, no drug placebo difference in either the carriers or the non-carriers. So that was the end of the bapineuzumab trial, although we did learn about one important side effect, which is ARIA, amyloid-related imaging abnormalities. And they frequently occur. This is swelling around blood vessels that occur when we give monoclonal antibodies. And we've seen it several times, and we worry about it. It's usually completely without symptoms, but it's not always without symptoms. And any symptom for a patient in a trial is very important to us. So the next step then, beyond bapineuzumab, was aducanumab. You wonder who names these drugs, right? Couldn't it be a little easier? Although you do see if it says MAB at the end, that means monoclonal antibody. So aducanumab, 2016, one of the drugs we're most excited about. So here we are with the CDR sum of boxes, a commonly used instrument. The left group of graphs on the left-hand panel show that at 6 months there was no drug placebo difference on this commonly used global instrument. All doses look exactly the same. But then look at the right-hand graph on the first panel. And what you see is that the placebo group in yellow has declined most. And with every dose, there's less decline. And at the highest dose, there is the least decline, and it's statistically significantly different than placebo. So we have begun to slow the progression of the illness as measured on this important global scale. Really important. And then to take an instrument like the Memantine, you see that in yellow is the declining placebo group, and in blue is the nearly stabilized high-dosed group. That's exciting. If we could keep patients at that high level of function, we would be very, very satisfied with that. What did the biology look like? Look at these scans. This is so exciting to me. So in the upper panel, I'm showing you, or in the left panel, I'm showing you at the upper part of it, 2 scans. And that red shadow is the amyloid in the brain that we think is the target of our therapies. And you see that upper panel, there's no difference between the first and the second scan. That's because that patient's on placebo, all right? But look at the lowest 2 scans, and you see a lot of red on the left and no red at all on the right. That patient's amyloid has been dramatically reduced by the monoclonal antibody, from abnormal to normal in terms of the amyloid burden of the brain. We have never seen a drug that would do this. And there's the corresponding clinical benefit. So we're pretty excited about this agent. It's still in trials. We're doing this trial now. So it reduced the cognitive decline on the CDR and the Memantine. I showed you that. It did not reduce the decline on the neuropsychological test battery. So that's a worrisome paradox in this clinical trial, because you really like to see consistency. The patients had prodromal or mild Alzheimer's dementia. So you see we're moving the populations that we're testing back to where they have more mild disease. We think it'll be easier to treat more mild disease. The patients were all biologically confirmed to have Alzheimer's disease by a PET scan, an amyloid PET scan. What a powerful technology has come in to help us with these clinical trials. And there was a high rate of ARIA. The phase 2 was small, 165 patients. So what could happen? What could go wrong with aducanumab? Well, a trial is most likely to succeed if you do exactly the same trial that just succeeded. So are there some differences between the phase 2 and the phase 3 trial of aducanumab that would threaten its future? Well, look at the differences. The PRIME study that I just showed you actually had 165 patients. They had intended 197. The 2 current trials, 1,350 patients. In fact, they just added 255 to the studies. The first study was done only in the United States. The second studies are done in the US and either 12 or 13 other countries. I've showed you the problems with that. The first study had prodromal and mild AD. The current 2 studies have only prodromal. The cohorts, there were 9 dose levels in the, there were 9 with drug and placebo in the first study and only 3 in the second study. The duration is different, 52 weeks versus 78 weeks. The primary outcome is different and the secondary outcomes are different. So it's very different. And these kinds of differences threaten the drug. It looks very powerful, looks very good, but I wouldn't quite put all of my portfolio trust in this drug yet. So after aducanumab, there was varabacestat. In fact, there were two drugs in 2018 that I'll mention, solanezumab and varabacestat. Varabacestat is the inhibitor of the other enzyme. All right, I said there are two enzymes that produce amyloid. One is gamma secretase, one is beta secretase. I showed you that we made patients worse with gamma secretase inhibition. What about beta secretase? Well, we finished a trial this year, almost 2,000 patients, mild to moderate AD, not biologically confirmed. The drug really worked. It produced a 65 to 90% reduction of the amyloid protein in the spinal fluid. How great is that? And it had no clinical benefit. That's a powerful trial because it shows that even if you do the pharmacology that you want, you achieve what you want, you may not get clinical benefit. That means, I think, that we are just not using either the right population, 65% is too much, or there was something unique about varabacestat. We'll have to determine that. But we showed that we can lower the spinal fluid A beta and not have a clinical benefit. Very important lesson. So the other drug of 2018 was solanezumab. I'm showing you on the left a drug placebo graph showing no difference. When they did a post hoc sub-analysis of mild patients, there was a difference in the mild patients compared to the moderate patients. So solanezumab was taken on the basis of a post hoc analysis from phase 2 to phase 3, and there was no significant difference between the two groups when treated with solanezumab. When treated with solanezumab. This was mild AD only. They were biologically confirmed by either CSF or PET. There was no benefit, no statistically significant benefit. You do see that the drug group is doing a little better. This is 11% better than the placebo group. It's not enough for approval and was not further pursued. So finally then, the last drug I'm going to present is BAN2401. This was presented for the first time at AAIC earlier this year. It's again a monoclonal antibody. It has a little bit different target than the other drugs like ADU we've seen. It targets the prefibrillar amyloid composite. They included in this prodromal and mild patients who had been confirmed biologically by amyloid PET. So you see these new technologies helping us to do better trials. This was a phase 2 trial, a large phase 2 trial, 800 patients. That's the way phase 2 should be done. It was an adaptive design with ADCOMS. So you haven't heard about ADCOMS before. We did include the ADAS-COG, but we based the calculation on the ADCOMS, a new way of looking at data in clinical trials. And so there was trial innovation. That's important. We shouldn't be continuing to do the trial that we did in 1992, right? We should be learning, and it looks like we are. But there was an irregular randomization caused by some regulatory concerns in Europe about the high dose. And this has introduced some questions about this trial. But overall, I believe this trial looks quite good. What I'm showing you here is the amyloid measurements done by amyloid imaging. You see they were all the same at baseline, and you see in the green line the highest dose that there were actually 81% of patients who converted from a positive scan at baseline to a negative scan at the end of the trial. That means enough amyloid was removed from the brain that they would no longer qualify for a clinical trial if they were scanned at that point. That's how much amyloid was removed. So 81% went from negative to positive to negative. And there was a low rate of ARIA, even in the E4 carriers. So this looks like something that's maybe safer than aducanumab. Here's the ADCOMS on the left and the ADAS-COG. You see, we're still using that ADAS-COG, and the ADCOMS depends on it. there was a 47% reduction in decline on the ADCOMS and a 30% reduction on the ADAS-Cog. So that's important. You know, that's a lot of slowing, and this is internal consistency, more than we saw in aducanumab. So we're pretty excited about BAN 2401. It hasn't gone into phase 3 yet. I believe it will be taken into phase 3. We'll see whether this triumphs. So we've got a lot of monoclonal antibodies in play. We've got solanezumab in a prevention trial called A4. We've got 2 other monoclonals I didn't talk about, gantenerumab and crenezumab. Both of those are in phase 3. I'm showing you a scan here from the gantenerumab early studies showing, again, dramatic reduction in the amyloid protein in the brain. So Gant looks like it's getting at the amyloid protein that we want. So let me conclude by some lessons that I'd summarize. One is, this is really hard work, right? This is hard. And we haven't won yet, but we believe we will. There's progressive improvement in understanding the biology of the disease. That's what Frank's work is so important. We're doing better at developing therapies and conducting trials, and you see the evolution of the measurements and of the tools that we're using, like amyloid imaging. We've learned that animals don't always predict the efficacy or safety. This was evident in the 1792 and the Taran-Flerbel studies that I showed you. We've shown that brain penetration, dose, and target engagement must be solved in phase 2. You've got to do that in phase 2 before you go to phase 3. Taran-Flerbel was a great teacher, the great professor of those lessons. And post-hoc subgroup analyses are a poor basis for guiding phase 3. We saw this in bapineuzumab and solanezumab. Biological confirmation of the diagnosis to ensure the presence of the target pathology, that is doing amyloid imaging or CSF to show that the patient actually has Alzheimer's disease. Very important. We know that we're wrong probably 30% of the time if we don't do that. That's 30% of people without Alzheimer's in an Alzheimer trial. It shouldn't happen. Global trials introduce heterogeneity of data. I showed you Josh's work on that, very important. We can make patients worse with some drugs like semaglutide. That's why we've got to be so vigilant about the safety. Treating earlier in the disease may be a benefit, although I think late-stage patients need treatments and we ought to be working on those populations, but we seem to be making more progress with early patients in ADU BAN 24, but not with solanezumab. And there are new trial tools that are emerging, like adaptive designs and the ADCOMS, that I think are going to be very helpful in the future. You see this with BAN 2401. So we've learned a lot. We've got a long ways to go, but I think we're really making progress, and some of these new drugs look great to me. I hope that this has been helpful for you in terms of thinking about clinical trials, why we should do them, why we have to do them, and what we've learned from them. Thank you.
We have time for a couple of questions. We have microphones, so.
They're rushing with the microphone there.
I have a question right here.
Where's that?
All right. Back here. Okay. The one study I think was varostasisat. Is that how you say it?
Varostasisat, yeah.
Yeah, that had the good penetration into the central spinal fluid, but it did not have any clinical benefit, if I recall.
Right.
The question is, can you use that drug as a transporter, like a molecule to transport other beneficial drugs that you're seeing to get better tissue or penetration? I don't know if that's the right question, but to get into the blood-brain barrier, to penetrate, to get into the brain?
Yeah.
Is that something that's being looked at?
It is something that's being looked at. Thank you for a great question. Varobasistat itself would not be a good transporter, but your idea is really a good one, and we are looking at other ways to increase penetration in the blood-brain barrier. There are many things I couldn't mention, but when you give an antibody, you get 1/1000th of the antibodies crossing the blood-brain barrier. So imagine how much we have out in the serum for how little we're getting into the brain. So if we could increase the brain penetration, particularly of a big molecule like the antibodies, we probably would be more effective. So we're looking at exactly that issue. There are several companies focusing on it. There's some exciting technology around that. It's a very central issue. Thank you.
First of all, I'll apologize for, I have no medical knowledge, so this is just what I'm taking from what you said. The scans that showed the reduction in the antibodies from the treatments or the trials, there was a difference even when you had good clearance in cognitive measures. So there's something, obviously something else going on. Do you want to speculate on why that happens?
Yes, yeah, really great question. Again, something I couldn't address. So we have hypotheses, right? And hypotheses are dangerous, but they have to guide our work, and unfortunately, hypotheses can be disproven. Here's the current hypothesis, is that amyloid is produced as one molecule by these two enzymes, and then it gradually clumps together, and then eventually forms a plaque. It is the intermediate clumped together form that seems to be most toxic to the nerve cells. So some of these antibodies only address the plaque, and we don't think that's very important. And some address the plaque and this intermediate form. And we think it's actually their effect on this intermediate form which is most helpful to the patient and most likely to reduce the rate of cognitive decline. So that's why we can move those plaques around without any corresponding clinical benefit. You were right. We did it with BAPI and we did it with 1792. Good observation. But we think we have an explanation for that. And we think that's why, for example, BAN 2401, which specifically targets those intermediate forms, late intermediate forms, may be a more beneficial molecule. One more?
You mentioned in one of the earlier trials that clinical meaningfulness required an improvement. Since we're identifying the disease earlier, wouldn't just keeping it at the same level also be meaningful?
I thank you for your question, and please write to the FDA with that thought, because we've had this discussion with the FDA, and you know, they're in a hard place, right? They don't want to approve a drug that doesn't really have meaningful benefit, and they've lived a long time with this idea of function being the measure of clinical benefit. They are yielding a little bit with these earlier patients, and in the most recent guidance, they opened the doorway to cognition itself being a key outcome. So it's not quite resolved yet, and I feel strongly, just as you articulated it, that keeping a patient's cognition normal should be sufficient for drug approval. But it's still a little murky from the FDA point of view.
Thank you very much.
Welcome.